We propose a method for regulating polymerase activity using artificial nucleic acid transcription factors. This gene regulatory architecture can serve as a building block for future in vitro genetic devices.
By tethering a DNA binding domain to a T7 RNA polymerase, this technique combines the scalability of DNA-based circuits with the functionality of transcriptional circuits.
Begin by making 9 dilutions of single-stranded BG oligonucleotide to a final volume of 50 microliters of double distilled water, from 5:1 to 1:5 ratios, as indicated in the table. Prepare 1 reaction mixture for each dilution by mixing 2 microliters of SNAP buffer with 4 microliters of BG oligonucleotide and 4 microliters of SNAP T7 RNAP. Prepare the RNAP control by replacing the oligonucleotide with double distilled water and the DNA control by replacing the SNAP T7 RNAP with double distilled water. Set up 11 reactions by adding 2 microliters of each sample to 4 microliters of SNAP buffer and 2 microliters of protein loading dye per sample. Heat the reactions for 10 minutes at 70 degrees Celsius before loading 2 microliters of each sample onto a 4 to 12% bistress protein gel for 35 minutes of gel electrophoresis on ice at 200 volts. At the end of the run, wash the gel with 3 water exchanges on a shaker for at least 10 minutes per wash, followed by nucleic acid staining with cyanide dye for 15 minutes. Image the gel on an appropriate gel imaging system and stain the gel again with 20 milliliters of Kumasi blue. After 1 hour, destain with double distilled water.